Cis-acting elements. DNA sequences close to a gene that are required for the gene expression, those sequences are called as cis-acting elements. Cis-acting elements. In case of eukaryotes, Tata box is called as cis-acting elements, while in case of prokaryotes, it can be at the position of -10 that works as an initiator. Proximal elements of promoter. In case of prokaryotes, there are sequences at the position -35 region which are called as proximal elements. In case of eukaryotes, they can be cat box or the GC box. In addition to this, there can be upstream promoter elements and downstream activating sequences. So these are all sequences which are called as cis-acting elements. In this diagram, we can see that here is the Tata box. This is the transcription start site and here is the Tata box. So this sequence which is called as Tata box is called as cis-acting elements. In the low diagram, we can see that here are the sequences, DNA sequences, which are called as enhancers. So these sequences, they are also called as cis-acting elements. What are proximal elements of the promoter? This is transcription start site. Here is the core promoter region where there is a Tata box. Here we can see that here is Tata box. In addition to Tata box, there can be regulatory promoter regions like the GC box. These are the GC box. Here we can see here is a cat box. In this diagram, we can see that cat box is here, cat box is here. So these are all proximal elements of the promoter. So they are called as cis-acting elements. These are actually those sequences to whom trans-acting elements bind. Proximal elements of the promoter. Here is the diagram in which there are two genes. This is gene A. Along with the gene, there is a promoter and there are enhancer sequence. In this diagram, we can see on the right part of the diagram, here is the gene B, here is the promoter, and here are the enhancers. So along with gene, there is a promoter region, then there are enhancers, and between these two genes, there are insulators. So enhancer 1 can stimulate the transcription of gene A. So this sequence that can enhance the transcription of gene A, while same is here that enhancer 2 stimulate the transcription of gene B. But there is an insulator between enhancer 1 and enhancer 2. So enhancer 1, because there is insulator, so this insulator, this enhancer have no effect on the sequences, on the gene expression of gene B. Same is here in case of gene A. These enhancers, they do not have the effect on the expression of the gene A. What are terminators? Are DNA sequences that just downstream of the coding segment of a gene, which is recognized by RNA polymerase as a signal to stop the transcription. So these are those sequences which are found towards the downstream of the gene, and here are the sequences which are recognized as a signal by RNA polymerase, and RNA polymerase that stops the transcription of the gene. So those sequences are called as terminators or terminator sequences. Enhancers are regulatory DNA sequences that enhances the transcription of the gene. Enhancers, usually they are stretches of 50 up to 150 base pair in length. Activities of many promoters are greatly enhanced due to the presence of the enhancers with those genes. What are silencers? A DNA sequence that helps to reduce or shut down the expression of a nearby gene, those sequences are called as silencers. In this diagram, we can see these sequences. Here there is a core promoter region. We can see that here are the sequences which are called as data box. Then towards the 5 end, we can see here are the DNA sequences which are called as enhancers. Then there is silencer, then there are enhancers. So silencers are actually the sequences that repress the expression of the genes. So the general binding factors, they bind to the region of data box, and the special transcription factors, they usually bind with the DNA sequences which are called as enhancers or silencers. Here we can see that insulator may block the enhancer action. If there is a DNA sequencer and insulator is present between the enhancer and the promoter, this insulator sequence that can block the enhancer action of the enhancer sequences. In this diagram, we can see that there is an enhancer due to there is an insulator. So insulator will act as a barrier for the expression of the gene, or it will be a barrier for the heterochromatin. So what are cis-acting elements? Cis-acting elements are all those DNA sequences which are called as data box, GC box. They are the proximal DNA sequences which are present to a nearby gene. They can be in the form of data box, GC box, cat box, particularly in case of eukaryotes, while in case of prokaryotes, they are called as -10 region, -35 region. In addition to this, in case of eukaryotes, there are distances, there are nucleotide sequences which are present at a distance. Those sequences are called as enhancers and silencers.